Okay, so in this session today, we will cover that part, explain why the cell are basic structure and about microscopes, and I will give hint about the cell structure and organelles, and we will complete that inshallah next session, right? While my explanation, I will focus on two things, right? The keywords. Okay, the keywords in our syllabus are يعني within two forms it will come to us. الفوكاب keywords and vocabulary يعني I have to answer using a particular word right يعني when I say this difference is significance you have to write there is significance difference. Don't say big difference, no, will not accept it if the keyword's not there, as if you did not write any answer. Okay. Also keywords include definitions, so definitions have to be memorized exactly as I'm giving to you, or as it given to us in the syllabus content, right, to be sure that you will get the full mark of that definition questions. Okay. About command words which will come in the question, I will usually I'm giving exam style questions. I will explain the command word for you and how you will answer. For each session we will have this type of explanation about command words of the questions which may come in the past paper related to the topic that we are handling today. Okay. This whatever we will cover in our session today, right? Now let's start. Okay, about the cell as a basic structure. Okay, I think you are familiar with that word. Cell is a basic structure of all of the living things, right? Living things could be, we learned that pretty well last year. We know some organisms are unicellular, other organisms could be multicellular. Okay, sorry, could be multicellular. Okay, right? So the common thing between all living things is the cell, whatever one cell or many cells. Okay, the cell must be there, right? How the scientists, or how the in the science, why how did they discovered cell? After the discovery of microscope, they started to examine thin slice of a plant tissue or animal. They found under the microscope these units which are repeated, regularly repeated for any part of tissue or any part of a plant. They found this is plant tissue. The same found when they are examined we're examining animals tissues also. This for plants, this for animals. Okay. With further examination of the cells under microscopes, okay, they found what? They found this structure which found in all living things. Okay, right? But during that time, they did not discover that there are organisms which are unicellular. They discovered it later, right? But at least they learned that there is a basic unit found in all of the living things, right? This basic unit must be surrounded with a structure. They called it a membrane or cell membrane. And that cell membrane contains jelly-like substances, and we call it cytoplasm or cystole. And each cell, whatever animal cell or plant cell, has tiny structure inside it. They called it nucleus. Sometimes you can see more organelles, sometimes not. It depends on the quality of microscope or the slides that they are examining. But at the end, they found the cells or the basic structure of all living cell, of all living organisms. cell membrane, cytoplasm, nucleus, right? And they called it a cell. Okay, so the cell is the basic structure, okay, of the living things. What we should call the nucleus or the structure which could be found inside these cells. These structures, you call them organelles. Organelles, what are organelles? Organelles, they are tiny structures found inside the cells. It could be surrounded by a membrane, maybe not surrounded by a membrane, right? Some of them had double membranes, okay? But anyway, they are structures found in each living cell to do particular function for the survival of that cell. Okay, and one cell, we learned it before, it can do excretion, can do respiration, can do division, all protein synthesis, all happen inside these cells. So if I want to define what is a cell, it is a basic structure and functional units found inside the living things. Okay, so if I would like to say what is a cell, the cell, okay, is a basic structure and function in all living things. Is it clear? Right? And inside each cell, there are what we called organelles, right? Okay, so if I would like to define the word or say what are organelles, okay, I will remove this one, right? I will write what are organelles. They are structures which found inside living cells to do particular function. Okay, so this is what they are learned about the cells and the beginning and the organelles. They give definition. They know there are basic unit which is repeated and found in all living things, regardless it's plants or animal. And inside each cell there are organelles which supposed to do particular function. And there are specialized cells, we know that from last year, right, which sometimes make difference in the contents of the cells. Maybe have some organelles disappeared or squeezed out of the cell during its specification. Some parts will be developed like cilia or flagella found in some cell, not in other cells. Is it clear? So if I will go to know about some keywords here, you have to learn what is the cell. As I said, cell is a basic unit of all living organisms surrounded by a cell surface membrane, contains genetic material and cytoplasm containing organelles, right? Okay, here they said genetic material, okay, not nucleus, because you know some cells will have nucleus like plant cell and animal cell, right? Okay, some cells will have nucleus, right, like plant cell and animal cell. Here is plant cell, here is animal cell. Some cells will not have any nucleus, have the genetic material like DNA found in the cytoplasm. Okay, so any cell, if I asked you define the word cell, you will say it is the basic unit of all living organisms which contain, okay, cell membrane, okay, genetic information, and don't say nucleus because there are some organisms like bacteria, right? Here, they don't have any nucleus, they have DNA free in the cytoplasm, right? And organelles. I told you organelles may be varied from one cell to another, and somehow any organelles will be there to do the function of that cell according to its speciality, right? Genetic material, yeah, it's supposed to be in all living cells, right? And cell membrane, it is a must. يعني if you remember last year when there is common questions usually come to in the part of cells, what are the common features of animal cell and plant cell? Okay, I told you cell membrane must be there, cytoplasm must be there. Okay, if the cell membrane is destroyed or ruptured, no cell anymore, right? Anything happened to the cell membrane that could destroy it, خلاص, the cell is killed, right? Okay, and if you remember when we were talking about antibiotic, how they are killing bacteria, we had hint about that antibiotic destroys the cell membrane of the cell, so the prokaryotic or the bacterial cell will be destroyed as well, because no cell membrane, no barrier between the surrounding of the cell, no structure which includes the contents of the cell and cytoplasm, so no cell then. You understand me? Is it clear? So the cell as a basic structure, okay, has cell membrane, genetic material, which found inside nucleus or not, and organelles. Is it clear? What are organelles? They are the function and the functionally and the structurally parts of the cell, right? Which responsible for the function of this cell and the life of these cells also, like ribosomes, mitochondria, nucleus, Golgi apparatus, endoplasmic reticulum. And in our chapter, we will learn a lot about the structure and the shape and function of the organelles which found in animal and plant cell, right? Nucleus, we will take it also in details when we'll go to the structure of the organelles, right? It is only found in eukaryotic cells, okay, and not found in prokaryotic cells. But let me explain this word. If I will divide this word into eu, right? Karyo, eu means true, karyo means nucleus. So the organisms which have a nucleus, we call them eukaryotic cell, eukaryotic cells. Let's explain pro. Pro means before. Eukaryotic cell, the organisms which have true nucleus. Prokaryotic, pro it means before or without nucleus, right? So the nucleus only found in the eukaryotic cells, not found in the prokaryotic cells, and we will take it in more details after a while. Okay, let's have a look to the cells which has nucleus or not having nucleus. Of course, this is a plant cell that can be seen under the here electron microscope. It has nucleus, and here is the nucleus, right? This is another animal cell, also under electron microscope, okay, right? Which we can see the nucleus clearly. And here the prokaryotic cell or bacterial cell, you can say, which does not have nucleus. Type. If I want to define the word nucleus to be clear for you, what is the difference between that one and these two? What is the difference here? If you notice here, the nucleus here, it is a part of the cell, or you can say compartment inside the cell. يعني the main difference between the eukaryotic cell and prokaryotic cell, here there is compartmentation. What does compartmentation mean? If you look at the cells here, there is compartment or a part of the cytoplasm surrounded by the membrane. It's called mitochondria, which is responsible for the aerobic respiration. Okay, there is compartment here which is surrounded by a membrane also, and this is responsible for photosynthesis, and which is called chloroplast. Here, a compartment which contains the genetic material, which is called the nucleus, right? Okay, the same compartmentation found here. There is part of the cytoplasm have single membrane. We call it temporary vacuole. Here is lysosomes. Here is mitochondria, rough endoplasmic reticulum. Here there is compartmentation, and I think it's very clear the cytoplasm has different compartments surrounded by membrane or even not surrounded by membrane, like that centrosome or centrioles, okay, called organelles. Here, nothing is there. You can see, right? Very few things. DNA naked in the cytoplasm, which is circular DNA, and short DNA molecule called the plasmid, right? And ribosomes. That's it. There is any compartment here, we can say it's mitochondria or even membrane surrounding the area which has the DNA to be nucleus, nothing is there. That's why it's called prokaryotic cells or before compartmentation and having nucleus. Okay, so if I would like simply to show you the structure of the nucleus, right? I will repeat it again when I go to give details about the organelles, but here is a compartment which has a double membrane. If the organelle has double membrane, we called it envelope. Yani this I will not say nuclear membrane, better to say nuclear envelope. Okay, it will be like that. Okay, nuclear envelope, which has also in between them nuclear pores. And if you remember last year when we were talking about the transcription, DNA, the mRNA will leave the nuclear through the nuclear pore to go to the cytoplasm. Inside it, there is another new organelle. We'll learn about it this year called nucleolus. And here the genetic information. And this simple structure, it's mentioned here clearly in the definition. Okay, a relative large organelle found in eukaryotic cells contain DNA of the cell that controls the activity of the cell and surrounded by two membrane which form the nuclear envelope. Is it clear? So here we should learn these two key words, which is eukaryote, prokaryote. What is the meaning of eukaryote? What is the meaning of prokaryote? Eukaryote organism whose cells or the cells contain nucleus, right? Its cells contain nucleus and other organelles. As I told you, there is compartmentation. There is compartmentation. Okay. In prokaryotes, no, there is no nucleus and no any other membrane bounded organelles. Is it clear? Okay. Shall I go to the next part, which we will go to the microscopes? Okay. I will go back to this slide one more time to show you this plant cell under electron microscope. And this is an animal cell seen by electron microscope. If I will draw or show you how the cell will be shown under light microscope, it will be very simple. Yani, the cell under electron microscope will be seen in that way. Cell membrane, I said it will be there. Nucleus will be seen by its envelope and pores. Nucleolus will be there. Genetic material will be there. There is structure, we learned about it last year, which is called rough endoplasmic reticulum, right? We have another one which we will learn this year, which is called smooth endoplasmic reticulum. We can see the mitochondria with the structures which found inside the mitochondria itself. Even the ribosomes we can see, and more details we can see Golgi apparatus, centrioles, many things. The same slide or the same cell, if I examined it under the light microscope, simply I will see very tiny structure with small dot inside it. Maybe I will see another thing, but all these details it will not be shown here at all. Cell membrane, yes, I can recognize it because it is determines the shape of the cell and separate it from the surrounding, so I can differentiate it from the surrounding, the cell membrane, and here is the cytoplasm, right? This is the nucleus. This is what I can see under the light microscope. Okay, so we can say here under the light microscope there is low resolution, and here under electron microscope there is high resolution or resolving power, we can say. But to learn what is resolution and why we can see more details here than here, okay, and also we have to know the difference between the two words resolution and magnification. I will make it clear after understanding what is the meaning of resolution. Okay, imagine with me, let's go here first. There is a thing or whatever, I don't know if you learn about that before or not. Okay, in physics there are waves. There are one whole chapter for physics, in physics, for waves, right? And we learn that waves, okay, have wavelengths, right, and frequency. And I think the wavelengths indirectly proportional with the frequency, right? Or I don't know, some books write frequency in the form of F. Okay, right? Okay, so if the wave will have long wavelengths, the frequency will be less. Okay, because both the multiplication of the wavelengths and frequency with Planck constant, it's equal the energy of the length. I think this formula, some of you will be familiar with it, right? Okay, يعني this is not يعني related to our syllabus, but I will explain only to understand what's going on in light and electron microscope fast, right? And if the wavelength is short, the frequency will be more, right? Okay, fine. This is the waves, and each type of waves has its wavelengths. You can see gamma ray, X-ray, some example of waves, and this is the range of the wavelengths of visible light, and these are the ranges of the wavelengths which we can see. يعني as my eye can see the least wave that found in the visible area of the light, it's 400 nanometer, and the highest is 700 nanometers. This is the ultraviolet, this is the infrared. Before more than 700 it will be infrared, this then for 100 nanometer it will be ultraviolet. Is it clear? Okay, so this is the wavelength of the visible light here. And sorry, you can see my screen? Do you see my screen?
Yes.
Okay, so this part I can say this is for the visible light. Okay, right? Because if we'll go to the name, it's a light microscope. So here it depends on the light, okay, to be magnified, to magnify the sample or specimen to be seen, right? This one is electron microscope. Okay, so it depends on an electron beam to magnify the sample or the specimen to be seen. Clear? Right. So here with visible light has long wave length, right? And less frequency compared to the electron beam. And this is the frequency and wavelengths of the electron radiation. Is it clear? طيب, how this will help? How this will help, right? To see an object or not, to see more details or not. Okay, let me show you here. Okay, imagine I have two objects. This one, okay, and this one. I have two objects. Object with very big size like that one, and another which is small like that one. Okay, and let's try the light radiation, visible light radiation, if it will be able to recognize it or not. First of all, you have to know we see objects by that way. The wavelengths will be reflected, right, back to your eye or lens to be seen, right? And I think according to the wavelengths, that object will be able to reflect the light, so that object will be seen by that radiation, right? Let's go to the other one. Yeah, the light can pass without recognized or without being reflected at all with these two small objects, right? So that's why we can see some things and we cannot see other things. Yeah, I think you heard about that some animals can see things we cannot see, right? Because maybe they have ability to see other radiation from the light like this and this, but only we are allowed to see between these two, between this area of wavelengths. Okay, that's why I think it's explaining now. If the object, okay, has if the object has diameter less than the 400 nanometer, or to be honest with you, less than half of the least wavelengths. Okay, يعني the object which can be seen by a light microscope, right? They have diameter, the minimum diameter, it's less than half, okay, of the shortest wavelengths. And actually, this, how can we measure the resolution of the microscope? But what is resolution, or what is the resolving power of the microscope? Resolving power of the microscope, it is the ability to distinguish between 2 points and see them as two different objects. Okay, يعني I can see this point from the object and this point from the object, right? I can see the whole object because it's able to reflect the light, so it can be seen. This one I can't. It's not even, I cannot feel or see even they are there because they are very tiny. They cannot be in the pathway of the light radiation. You got it? Is it clear? Right? So if I want to measure the resolving power of the microscope, it is half of the shortage wavelengths of the radiation, right? So if I would like to say what is the resolving power, right? Okay, I can measure it, or I can say it is the half, okay, of the shortest wavelengths of the microscope. The resolving power of a microscope, it is the half of the shortest wavelengths of the radiation, half of the shortest wavelength, right, of the radiation used. Type, let's go to the light and see where is the shortest wavelength. Huh? Where is the shortest wavelength? 400 nanometer. So half of the 100 nanometer, okay, it will be what? 200 nanometer. You got it? So the resolving power of the light microscope is 200 nanometer. Is it clear? Huh? Is it clear? So the resolving power of the microscope, of the light microscope, is 200 nanometer. Is it okay? Type. What about the electron microscope? Here I said electron microscope have very short wavelengths, and consequently it will have very, very high frequency, right? So if I will tell you about the resolving power of the electron microscope, it's 0.5 nanometer. Okay, so the resolving power of electron microscope equal 0.5 nanometer, and the resolving power light microscope equal 200 nanometer. Is it clear? And you have to memorize these two values. You have to memorize this part. What else I should memorize in that part? I should memorize the keyword or the meaning of resolution. In all of our session, the keywords all the time I will give it to you in that side of our board, right? I will send you all of this classwork, and all of the keywords will be given to you in the right side of the notes. Clear? Okay, so resolution is what? Ability to distinguish between two objects which are very close together, right, and see them in as a distinct 2 points. Okay, and the higher the resolution of an image, more details we can see. Is it clear for all of you? Right? So in that part, I have to learn about, okay, the resolving power of each microscope, right? Okay. Second, about the resolution. Okay, definition. Type. What is the difference between resolution and magnification? Simply, I will tell you what is difference between resolution and magnification. But let's have a deal first. Resolution, we can see more details. I can see things here, never I will be able to see if I will use a light microscope. Okay, type. What is magnification? Magnification, it means enlargement of the size. Like, you can see that cell? Yes, we can see. Okay, if I enlarge the size of that cell as much as I can, do you see more details? No, only the size of the specimen become bigger, but no more details. I can see it clearly, but nothing will be added up to the image. Is it clear? Time. Here, magnification, it is enlargement in the size, right? Okay, without saying more details. But resolution, no, more details, more resolving power of the microscope, more details I can see. You understand? Okay, so also I want you to learn the difference between, okay, magnification and resolution. Okay, magnification, how many times the image is larger than the actual size, and we learned last year how to calculate magnification, how to calculate actual size, and again we will take it this year in the practical, inshallah. Is it clear? Okay, let's cover one more time. what we had finished here. We learned about light microscope. It's type of microscope which is depending on light radiation, electron microscope. Another type of microscopes which depending on electron beams to magnify the image of the sample, and it has very high resolving power, right, which enable us to see more and more details inside the cell. Even we can see the organelles which in nano diameter, like microscope, which is from 18 nanometer in prokaryotic cell and 22 nanometer in eukaryotic cells, right? We learned about the resolving power of each one, and you have to memorize the value. In light microscope, there are 200 nanometer. There is 200 nanometer resolving power. In electron microscope, there is 0.5 nanometer resolving power. You have to know clearly what is the word resolution mean and what is magnification means. Is it clear? Okay. طيب. Let's go to exam style question, or question usually come to cover that part of resolution. For magnification, questions will come. You will measure the size of the image, calculate actual size or the opposite, and we will cover that in the practical as well as paper one practicing questions. But there is a question usually come to cover that part to compare between the two microscopes regarding the point of the resolving power or resolution, right? Let's write the question here and let's see who will be able to answer it with me, okay? That question, it's repeated usually in the past paper, right? The question will, the question will say and ask you, for example, why we can see ribosomes using electron microscopes, but we cannot see ribosomes using the light microscope. Or why we can see crestia inside the mitochondria using electron microscope, but we cannot see the crestia inside using the light microscope. You got it? Okay. The style question. Okay, explain why ribosomes can be seen using electron microscope, not light microscope. This question coming for 2 marks. Okay. How can I answer that question to secure the 2 marks or even 3 marks? You know, if you can answer first, I will give you try to answer. Then I will give you the model answer. Who can answer that question? I will give you like 1 minute to write the answer in the chat. Then we will discuss your answers together. Then we will give the model answer. Okay, Adam Hani. Adam Hani, I said if we will make the image bigger, yeah, we cannot see more details. Excellent. Okay, but you can use one more word. Okay, magnification will not work, but what is the word which you should use? I think resolution. Better to say light microscope has low resolution or less resolving power compared to the electron microscope. Good, Mariam. High resolution than light microscope. Abdullah Omar, more resolution than light microscope. Yes, but if I will say that, and my advice also to write, mention the value. What is the benefit if I will memorize the resolving power? I will not use it in my answer. Say it clearly. the light microscope has a resolving power 200 nanometer. Okay, the electron microscope has a resolving power 0.5 nanometer, right? Okay. First of all, you can say define the word resolution. We can define resolution and mention the resolving power of each microscope, right? And later we will learn the diameter of the organelles. So I will know that ribosomes has diameter 22 nanometers. This is the diameter of ribosomes, right? If I don't know the diameter of the organelle, simply I can explain it, right? How can I explain? I will say ribosomes diameter is less than the resolving power of the light microscope, as I explained here. Imagine with me, these are ribosomes. Its diameter less than 200 nanometers, so no way to be seen under light microscope. And its diameter, which is 22 nanometer, is bigger than 0.5 nanometer, the resolving power of electron microscope, so it can be seen clearly using the light, using the electron microscope. Is it clear? Okay. So the answer, whatever we will write now, okay, it will cover even 3 marks, not only 2. So here I will write, okay, the microscope equal 0.5 nanometer, and the resolving power of light microscope equal to 100 nanometer. Okay. If you know the diameter of the organelle, simply if you mention the diameter of the organelle, you will take the mark. Okay, but I will write in explanation in case if I don't know the diameter of the organelle, so you can explain, you will get the full mark, right? The diameter of the ribosomes okay is less than the resolving power of light microscope, which is 200 nanometer, and okay, more than the 0.5 nanometer, okay, which that's why it can be seen only under light microscope. So it can be seen only using electron microscope. You can add one more point. You can also define resolution. So this will be 1 mark. This will be 1 mark here. And the explanation here will be the other mark. A third mark if you would like to take, if be more than 2, define resolution. Right. There are type, still up till now we're still using the light microscope as well as electron microscope. If electron microscope will give us a lot of details, we can see a lot of things inside the cell. Why we still keeping the light microscope? Why we still using it? Because each microscope has its own advantage, has its own disadvantages, right? Let's go to take more details about the 2 microscopes, right? From these details, we will learn how to compare between them, right? Because sometimes questions will come to compare between them, and advantage and disadvantage of each one also we need to learn about that part. Clear? Okay. طيب. I will divide my board كده into 2 parts. I will write here light I will write here electron microscope. Here the feature of comparison. First of all, what is the source of radiation? Fine. Type. Simply how the light microscope is work. The light microscope, if I will go here to the image, this is the source of light. It's here. And here there is the object, the specimen. I will put it here in the stage of the microscope, and you're supposed to work on that in the lab. And here there is the lens or objective lens, which receive the reflection of light from the sample. I will select one of them, not all, and each one has different magnification power. And here the eye lens at which I will look at my sample and examine it, right? So here focusing of the light on the sample, magnification twice by that lens, and that lens here, the specimen can be seen and I can use. And you can see I can prepare a slide by myself in the lab. This part of your syllabus will learn you how to prepare simple slides. Right, we will put this slides here, okay, and I can see using light microscope, okay. It does not need an expert to prepare the slide. That's why we will learn you how to prepare it this year. What about electron microscope? You can see this is the electron microscope, and this is the expert who's supposed to prepare the slide. Not anyone can do it, okay. And imagine the lens here is metal, right? It's working by, yani, we will put heavy metal exposed to a very high temperature or something which will make excitation for electron, which will pass through metal lens. That metal lens will hit the sample, right? And electrons which is reflected from the sample, it will be received by X-ray film or screen like that. Because I will not put my eye in front of beam of electron. I will lose my eye, right? So here I will receive film, okay, of the sample image on X-ray. And most of us saw X-ray film which used for scanning of points. It's white and black image, right? Okay, and if you see any image of electron microscope and colored, it's false colored image, not real colors, because the actual image which will be taken by electron microscope, it's black and white X-ray film. But light microscope, no. I can prepare the slide, I can stain it with different stains, I can see colorful slide. I will ask you a question. If I want to examine live sample, and I want to see the cells during the process of mitosis, I want to watch the process, which microscope I can use, electron or light microscope? To see live sample, yes, because only light will go through the slide, so it will not kill the cells. But when you hit the cells with beam of electrons, right, the sample will die. So one of the most important advantages of using light microscope, we can see live samples. I can see mitosis, meiosis, I can see microbes, cell binary fission for bacteria. All I can use using the light microscope. But electron microscope, no, it's dead sample. And actually no way to have the similarity of dead and life things, right? Yeah, and some changes or some little differences between dead and life sample will be there. You got my point? Hmm? Okay, so here we learned two good advantages about the light microscope, right? First of all, it's easy to prepare the sample, does not need an expert to prepare it. In light microscope, I can see colorful specimen, I can see. In light microscope, I can see live samples or live slides. You got my point? Okay. So up till now, electron microscope has one advantage, which is high resolving power, right? Light microscope has high, sorry, it is easy to prepare the sample. I can examine colored specimen. I can see live samples, right? And also, which one is smaller, portable, light microscope, cheaper, also light microscopes. That's why most of the university laboratory and schools laboratory has light microscope. Maybe the whole university will have one electron microscope, right? It's a big, need space, expensive, need expert to prepare the sample. Not anyone will be allowed even to use it. Let's go to our comparison one more time. So here the source of radiation is light, okay? Here it is electrons beam or electrons. Here the lens, it could be glass or plastic. Here it's metal, made up of metal, okay? Here image, here it's colored, here it's black and white. Here it's life or dead. Here it's only dead sample. Type. Here slides, easy to be prepared. Here need an expert. Okay. Here price, here size. This one is expensive. This one is cheaper. Size here portable, small. Here it's need a huge space. It's large, very large. Yani it could be found in one room for electron microscope, occupy more space. Is it clear? Okay. If you have your notebook, I would like you to draw that table and complete it. Then we'll discuss your answer together. Or you just draw the table and fill the blanks, right? To discuss it together. So here we learned about microscopes, their resolving power, what is the difference between resolution and magnification, and also we learned about the keywords which we will need here about the cells. What is the cell? What is the definition of the word cell? What's organelles? What is nucleus? What is eukaryotes? What is a prokaryote? And what are the types of microscope which we can use to examine the cells, right? Okay, there is a practical part related to the microscope, which is talking how can we measure the actual size of a sample using the microscope. And again, we will revise the part of magnification, how to calculate magnification, how to calculate actual size. We will take it, inshallah, this week on Thursday, right? To learn you about that practical part before practicing questions which related to the part that we finished, right? The next part of the topic, and I would like to start today to talk about the organelles. يعني I already started about nucleus, so only I will complete the structure of the nucleus and the function. And inshallah, next session we will take more details about each organelle and structure found in the cell, right? So يعني only quickly كده I will mention about the organelles and what we should learn about them. Just a second. طيب, the next part, as I said, we should learn about the organelles. طيب, what I should learn about organelles. What I should learn about organelles. Huh. Number one, we should learn about the shape of the organelle. how it looks like. Okay, continuous membranous structure. Okay, it has oval shape, rod shape, right? Rounded under the microscope, spherical shape. Okay, why I will need the shape? Sometimes they will ask you to describe the shape, right? Or also to recognize it, because a common question will come, they will give you the image and ask you to label the nucleus, label the mitochondria. So if I will know the shape of the organelle, how it looks like under the light and electron microscope, I will be able to answer the question, right? Number two, okay, structure. And under the microscope, I can see the nucleus has rounded shape, or if it's 3D, I can say spherical shape, nucleolus, okay, sided, there are details, this is the shape. But the structure, I have to mention, it has double membranes called the envelope. It has nuclear pore. It has nucleolus. It has genetic information in the form of chromatin. You understand? So structure, you will describe the details, the contents of the nucleus. Okay, and we have to learn about the function. What is the function of the nucleus? It contains nucleolus, which is responsible for making ribosomes. It contains chromatins, which control the activity of the cell by controlling the protein synthesis. Okay, cell division, right? It carries the genetic information of the cell. And the fourth thing that we have to learn about any organelle, diameter. So this what we will learn, huh? Next session, inshallah. Okay, about organelles. We will discuss each organelle, the shape under electron and light microscope. I will show you the image, the structure of each one. How can I describe the structure? Okay, because one question came, describe the structure of the nucleus. And also we should learn how to draw the organelles, because some questions will ask you to draw, complete the drawing, or draw the organelle. Okay, and the function of each organelle, diameter of each organelle, and how to draw each organelle. I hope it's clear. And our session for today is finished.